My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to Snake Talk Rewinds. Today we're doing something a little bit different that we'll do periodically, but we're going back into the archives and we're going to pull out some of those episodes from the past that you all really enjoyed and just give you another opportunity to listen to them. So today we're bringing you one of our classic conversations, an episode that remains just as fascinating and relevant today as when it first aired. In this one, I sit down with Dr. Stephen Beaupre from the University of Arkansas. He's one of the world's leading experts on timber rattlesnakes. Together we dive deep into timber rattlesnake biology and talk about how what we've learned can guide effective conservation. So whether you're hearing this episode for the first time or you're revisiting it as an old favorite, you're in for an in-depth look at one of North America's most iconic snakes. Let's dive in. Here today talking with Dr. Stephen Beaupre. I've known Steve for probably 15 or 20 years now. I met him a long time ago, it seems like, when I was doing my PhD out at Idaho State University. And my advisor at the time, Dr. Chuck Peterson, was close colleagues with Steve, and I had the opportunity to meet with him and interact with him at a variety of conferences, which was really great and really helpful to me because while I was doing my PhD, I was really interested in rattlesnakes. I've always been interested in rattlesnakes, and I was really interested in particular how changes on the landscape were affecting rattlesnake populations through the prey that they were feeding on, through the mammals that they were feeding on. And Steve just happened to be, probably the world expert in that general arena of rattlesnakes and energetics and life history. So I have a great deal of respect for Steve. I'm very thankful for some of the advice that he gave me in some of the discussions we had years ago. And I'm really glad to be talking to him today. So welcome, Steve. It's great to have you here.
Chris, it's wonderful to see you again. It's been a long time, and it's funny. I'm listening to you, relate the story of how we met and interacted, and I'm thinking the same thing about your advisor, Chuck Peterson, who I met when he was a graduate student and I was an undergraduate at University of Wisconsin when he was visiting Warren Porter's lab, and I had the same relationship, and I have to say that it goes both ways. I have always enjoyed your work, and you're we're sort of thinking along the same lines in a lot of ways, and I really appreciate that, and I know how hard it is to do the kind of work that we do, tracking rattlesnakes in rough country, and it's a commitment that comes from the heart, and I appreciate that a lot.
Yeah, that is the truth, and it's just amazing these connections you see through the snake world and snake biologists and conservation biologists. So it's great to hear about you and Chuck. Why don't you just give the listeners just a fairly brief introduction to who you are and what you're currently or what your current position is in your career.
Right. I came to the University of Arkansas in 1995 after a postdoc, and I think I maybe tell you more about that in a little bit. But so I started as an assistant professor in 1995 and worked here through the normal procedure of becoming an associate professor and a full professor. And then I got sucked into administration. I was a department chair for 4 years. And then after that, I became an associate dean. And I've been an associate dean since 2016. And it's been rewarding in a lot of ways. And one of the reasons it's been rewarding is because they've made a deal with me that allows me to continue my research while I'm in an administrative post. So I do a little bit of teaching, and I've still got a full and very active lab. But most of my days are taken up with personnel issues and logistical issues, and lately COVID-19 issues. So that's what I've been doing, and hopefully, another few years, and I'll go back to the department to finish up my career and finish up some publications I've been working on, and drift quietly into the night.
Well, that's great to hear that you've been able to maintain your research program as your career's grown in administration. That's seems like probably a pretty rare thing. Maybe it's becoming more common these days, but that's really great that you've been able to do that. So one thing I always like to ask our guests are how they got into snakes, because if you look at a segment, a cross-cutting of the general public, snakes and a deep interest in snakes is a very kind of eccentric thing. Maybe not to me and you, but to a lot of people.
No question about that. Yeah.
I just like to ask people, and I mean, you can go back as far in your history or childhood, or maybe it's something more recent, but what are some of those events that just really got you interested in this amazing group of animals?
Well, for me, it's a really easy question because I remember exactly when and the moment that this whole thing started. I was 5 years old and walked out. My family lived in Wausau, Wisconsin, which is not a particularly snaky place, but there's a fair number of interesting species there. And I was 5 years old. I didn't even know what a snake was. I walked out the front door. We had a perennially wet, dry ditch in the front that was full of leopard frogs. And I walked out there one day and was watching the water go by and saw rustling in the grass. And it was a garter snake, good sized garter snake that had a frog halfway down his throat. And I mean, literally the legs of the frog were sticking out in this crazy looking, striped critter. And I was, instantly fascinated and just couldn't believe my eyes. And, so from there, I rapidly got to the point where I could say the word herpetologist, and that was sort of my life goal after that. And my parents went through insane times with me bringing, snakes home and putting them up in cages that weren't always secure. And the story. It's like you get obsessed and you can't help yourself. And it's like every chance I got. And, most parents, when they do activities with their kids, they take them to the ballpark or whatever. My dad would give up weekends of golf to drive me out in the middle of nowhere and just let me run. He would sit in the car and read a book while I went up and caught snakes. And just quite a commitment on his part to let me explore that. So that's really how I got started with it. And then there were a number of people along the way that sort of fed that fascination and kept me moving. And I would have to admit that there are times where in life I walked away from those goals and usually got pulled back into it again with the right mentor who said, you can do this. You can make a living out of this, which was a shock and a shock of amazement to me.
Yeah, that's great. First of all, that your parents were supportive, even if they didn't share the same fascination. Allowing you to have these animals in the house, and even taking you out and sitting there and reading a book, as you described. It's, as you're probably well aware, there's a lot of, or some research around how much of this fear in snakes that a huge segment of humans have, how much of that is kind of genetic versus behavioral, or something that we've learned. And we talk about that. We'll be talking about that more on this podcast with people like Harry Green and some others. Mm-hmm. And so anyways, it's just fascinating to hear a story where your upbringing, your family was very open to that. They didn't teach you to hate and that you had to kill any of these animals that you saw.
Yeah, that's fascinating, isn't it? I think one thing I've always noted is that no one is almost no one is neutral to snakes. You're either absolutely fascinated by them or you're absolutely repelled. And it's not a 50-50 ratio at all.
Yeah.
So I rarely met anybody that was actually neutral to them. And my current wife might actually be an example. She doesn't fear them, but she doesn't particularly care about them. But I shouldn't say my current wife, my wife. Okay. I'm gonna get in trouble for that later.
You have plans for the future, Steve?
Yeah. No, we better edit that part out. That's gonna just get me in trouble. It's one of those things you can't take back,
Yeah, no, that's great. But well, okay, Steve, that's a great segue. I'd like to talk a little bit about your career path and how you ended up where you're sitting today. So it's it's a difficult field to make it in, whether you want to be a wildlife biologist, whether you want to be a herpetologist, or even more specifically, a snake biologist of some type. So I'm just curious, you talked a little bit about kind of your upbringing, but what was kind of the career trajectory, maybe as you went through college and graduate school and postdocs that brought you to where you are today?
Yeah, so with the interest that I had as a kid, getting into high school, I took every biology class I could find, and of course, at the time it was like, yeah, of course I'm going to college. I didn't really have any other options, and so I got out of my high school, went to the University of Wisconsin in Madison, and declared a major in zoology, and that all went very well. And I got to the end of it, and I was like, well, what the heck do you do with a major in zoology? Well, I guess I could work at a zoo, right? Yeah. Now, well, that sort of didn't. The last year or so, I got signed up into a seminar class that Dr. Warren Porter at the University of Wisconsin was teaching. It was called Biophysical Ecology. And I took it because it sounded interesting and fit into my schedule. And it turned out to open some amazing doors for me. It was a basic course. It wasn't basic. It was an advanced course about mass and heat flow through animals. And it was a very engineering sort of oriented course. And meeting Warren, he's like the world's best cheerleader. He was always pushing me to think more and to engage more in it. And the result of that was that I ended up working as a field hand for him and some of his graduate students for a couple of summers after I graduated. And there was a point, we'd worked in a lot of work on small mammals in Alaska and in Colorado. And there was a point where we were having a conversation and I told him about my interest in herps, and he knew about that because I've been catching timber rattlesnakes, through the whole thing. And he said, you can do a master's degree and try a career on this. And so I did. I just said, okay, let's give it a shot. Spent 2.5 years in a master's program with Warren Porter. And then right at the end of that period, I met Art Dunham, his wife, Karen Overall, Dr. Karen Overall, who's a veterinary medical doctor and a PhD, came to University of Wisconsin to work with Warren Porter on her PhD. And so the two of them came and spent a sabbatical. Art spent a sabbatical. We ended up sharing an office. And when I started talking to him, he started talking about life histories and, telling me his perspective on all that. And I just got absolutely fascinated by it. Went to Big Bend and spent a summer working with him. And then at the end of that summer, I started as a graduate student at Penn and in his lab. And I have to say that, this was touch and go. You say it's a difficult path and there's no question about it. I sat there and I thought, I suppose if I screw this up, I can always become a banker or something, but bottom line was I showed up to Penn and got into those graduate seminars. And when the first 2 weeks, I'm thinking, oh man, I am so outclassed. These people are so much smarter than me. I am not going to make it.
I just wanted to take a break and tell you guys, if you care about snakes, wildlife, or wild places, and you like what you're hearing on this podcast, go to www.orian.org now to make a difference.
Yeah, so when I went to Penn as a new graduate student in a PhD program there, I spent, a few weeks in the seminars with these advanced students, and I was just blown away by how smart they were and the depth of their knowledge and their critical thinking abilities. I thought, I'm never going to make it. I'm not like this at all. I don't have a chance. I spent a couple of weeks thinking, should I leave? What should I do? And I finally decided, there's a little flame in the back of my head that says, I'm not going to leave until they tell me I have to. I'm not going to give up. I'm not going to leave till they tell me I've got to go. And that's been my motto ever since. Whenever I get up against something goofy, I think I'm not leaving till they tell me I have to. And, that's worked for me. And it worked for me through my degree. And, after I got through with my PhD, which was a lot of hard work,
and your PhD, is that when you were working in Texas?
Yeah, I was working in Big Bend National Park studying rock rattlesnakes on an elevational gradient and applying some of the life history concepts that Dunham has written extensively on. And it was a lot of hard work. I went through 2 heavy lug vibram sols a year, basically, walking rocks out there. And there were good years and bad years, but and there was a lot of intellectual development too, a lot of development of critical thinking ability and stuff like that. And by the end of the 6-year period, I produced a dissertation. And by the way, this is kind of the funny part. My committee members would often tell me, "You can't do this. There's no way you can accomplish what your goals are, and you need to change your project." And I thought, you know what? I'm not going to give up. I'm going to keep going until they tell me I have to leave. So at the end of the day, I probably didn't get as much data as it would have been, ideal. But I got enough to get a dissertation and keep moving on in my career. And that's, you look back on it and say, I could have probably chosen a better system, but there were other constraints and other issues ongoing. So in the end, I finished a PhD, and then that very next year got a postdoctoral position with it was co-advised by Dave Duvall and Harvey Poe at the Arizona State University West, which is a small campus up in Glendale. And we had a really neat study system out in the north of Scottsdale in McDowell Mountain Regional Park, just Western Diamondbacks all over the place, and a fairly high density of Scutellatus, of the Mojave rattlesnakes as well. And had just a ball marking, capturing, and radio tagging those snakes to try to learn more about their energetics and social interactions. And that lasted about a year and a half, almost 2 years. And the whole site was destroyed by a major fire in July of 1995. And it just so happens that that was my transitional summer. I had gotten the job at Arkansas and started in 95 in Arkansas. And that kind of brought me back to the timber rattlesnake system. One of my major concerns about being able to work in a given area near a university was whether I could find snakes in the area and actually find something that was workable. And it just turns out that the timber rattlesnake, which is a snake that I grew up with, was the best candidate in terms of density and the right body proportions to keep working on the kinds of questions I was interested in. And so that's what I did.
That's great. Great trajectory. And it sounds like you had a lot of good experiences in the field in Texas and other places.
Incredible.
So let's shift gears now, and I just want to talk a little bit about timber rattlesnakes, and we'll start off by kind of going through just some of their biology, and I'll ask you a variety of questions there, and then we can dive in a little bit deeper and talk about your research program over the years and the things you've been working on and learning. So First of all, I'll tell you that timber rattlesnakes, I like you also grew up in a kind of a northern climate. I grew up in New England originally, where we had, venomous snakes, first of all, were rare, but the one that we did have was the timber rattlesnake.
Uh-huh.
And they've probably are, if I was cornered and had to pick a favorite snake, the timber rattlesnake would be it. And it's been really nice to get back east and be able to work on them again. They're also just, I call them, they're kind of America's snake, if you will, in that, the majority of humans in the country overlap with the range of the timber rattlesnake. So many of the kind of human snake, venomous snake interactions that happen in the country occur with this species. And then there's also just such an interesting and fascinating history in terms of human history and how we've used the timber rattlesnake as a symbol going all the way back to people like Benjamin Franklin, and it's just a fascinating history. So it's probably again my favorite snake on the planet, and I find their biology just fascinating. So I guess the first thing I would ask you is, where do you find these snakes? So if you were to describe to our listeners kind of their distribution and maybe in a very general sense, why they're distributed in those areas, what is the distribution of timber rattlesnakes?
Well, you kind of already mentioned the general distribution of the animals. They go all the way from the southern part of Maine down into central Florida, and then across all the way into Texas and up through Oklahoma, Kansas, and over into eastern Minnesota and the Great Lakes region. Historically, they popped all the way up into Canada, but most of those populations, I think all of those populations have been extirpated at this point. In terms of their more focused, let's say, micro distributions, the really cool thing about timber rattlesnakes is that they're not hugely polytypic. They're really, in other words, they look about the same. You can recognize one wherever you run across it, because there's not a lot of geographic variation in their patterns and colorations. There's certainly some, but it's not very consistent, and it's all part of sort of one big species that looks about the same everywhere. They get bigger in the southern part of the region in general, but there are some interesting exceptions to that. And you typically can find them, depending on where you are in the range, in a variety of habitats that ranges from swamps and river bottoms to upland rocky bluffs to dry prairies to swamps in Arkansas, for example. We get them down there. Any uplands, and the further north you go, the more tied they typically are to rocky outcrops that can allow them to get down into crevices that go below the frost line. According to our research, they are not freeze tolerant, so they will not allow themselves to drop below freezing in the winter. So they go deep enough to where they reach the annual mean temperature of the area and then winter in that kind of an environment, and then emerge based on some as yet undetermined cues in the spring when the weather warms up again. But in general, it's interesting as the habitats dry to the west and get more prairie-like. We have some really interesting populations that are out in what you might consider open prairie. And our populations in Arkansas are a mix of populations in open habitats and treed-in habitats. And a lot of that might be historical, because the treeing of the landscape has changed dramatically over the last 100 years in Arkansas.
That's fascinating. So across the timber rattlesnakes range, the area I have the least experience with them is certainly on the western portion of their range. And I've always kind of thought in the eastern portion, if you will, so think like New England down through the Appalachian Mountains and down into the southeastern coastal plain, I've always kind of thought of them as somewhat of a hardwood forest associated animal. And I say that in a very general sense. So obviously through the Appalachian Mountains and the northern hardwood forests. But then even as you get down into the southeastern coastal plain, where you primarily find them are in these bottomland swamps, as you mentioned. And those are the places where you have the hardwoods, where the pines are more in the uplands. And don't get me wrong, you can find a timber rattlesnake up in these pine forests in the more upland setting, but you certainly see them more in the swamp. So I'm curious, as you get into that western part of the range. You're talking about these snakes out on the fringes and into the prairies. Are they also following kind of riparian hardwood areas? Or are they out in the open, say, tall grass prairie?
Well, let's see. That's a great question. I haven't actually looked at the relationship between their western distribution and proximity to drainages or river bottoms. My impression of, populations that are out in Kansas, that Henry Fitch worked on and some of the folks that have worked up around Lenexa lately, that these are rock outcrops that occur in what really amounts to open prairie. One of my graduate students, worked in a system up in Missouri, Rod Wittenberg, and he worked in this really interesting place where there was an old quarry in the middle of an agricultural landscape. And the snakes used the quarry for denning and gestation, and they moved out into the agricultural landscape and used fence rows mostly for foraging. And they took a large portion of voles and squirrels and chipmunks, but voles made up a greater proportion of their diet, I think. And that kind of the way I look at these animals is they can persist just about anywhere as long as they have abundant food, and as long as they don't have a major source of mortality like a highway, bisecting their habitat that produces a large amount of road mortality. So if you feed them, they do just fine.
Yeah. Huh. Speaking of feeding, one of the topics that a lot of the kind of lore around this animal and all venomous snakes, where it comes from is the concept of venom. And you mentioned feeding, and certainly in rattlesnakes, venom is adaptation for feeding. And so I'm wondering if you just take a couple minutes and talk about venom specifically in the timber rattlesnake, everything from, how is it produced to what it's used for, why do these snakes have the venom, and how it might vary, say, across the species, those types of things.
Yeah, so let me start with a disclaimer. I am not a venom biologist, not a venom expert at all, and most of what I can tell you on this I would have read by the work of other people, especially people like Steve Mackesy. So I look to guys like Steve to keep me informed about venoms. What we do know about the really interesting thing about being a snake is that you have no legs, right? And that means that anytime you're going to feed on something, you have to catch it with your mouth. And that, I don't know if you've ever tried to catch a squirrel in your mouth, but it's really, it takes days, and it's not of good taste when you get there. And but as a snake, the question is, how do you deal with that? Well, one way you can deal with that is in a feeding mechanism sort of way is to only feed on things that you can physically overpower with your jaws and your mouth, right? The other thing is you might have another method to subdue larger prey to increase the meal side, and constriction is to increase the meal size. And constriction is a good way that a lot of snakes use to accomplish that. But somewhere along the line, evolutionarily, venoms evolved, and venom delivery systems co-evolved in a much more functional direction. And the interesting thing about that is that if you look at the pit viper skull, for example, it's what we call a very kinetic skull. The bones are not fused together in general the way they are in a human skull. So, for example, the joint between our lower jaw bones, right up in the middle where your chin is, that's called the mandibular symphysis. And in mammals, that's generally fused into a single bone, and it's very tough. In snakes, it's separate bones, and they're attached by ligaments. And also, the other elements of the skull are not fused because they allow independent movements, for example, of the jaws and the different rows of teeth. And that's all useful for dragging a very large item down into your throat. And so one of the things that comes along with venom specialization and delivery system specialization is a lightness of the skull and a kinetic, nature to the skull. Now that helps in two ways. One is, you can produce mandibles that swing forward. You can have very long fangs that deliver toxic venoms that swing forward and fold back when they're not in use, so you don't poke yourself, And the other thing is very helpful for is if you're going to be doing things like eating things that are bigger than your head. I've tried, eating things bigger than my head, and I've always had to cut them up into smaller pieces. It's really frustrating. But just to get a feeling, a squirrel, in our timber rattlesnakes, a medium-sized male or female can take down a squirrel, and it might be close to 100% of that snake's body mass. And when you look at even young snakes, they can take food items up to 150% of their body mass. So they're phenomenal at taking very large meals, but they only do it once in a while. And a snake in our system, based on the energetic modeling we've done, can take an adult male rattlesnake can take a single gray squirrel a year and meet its entire annual energy budget with one meal. So that's pretty cool. Now back to the venom, of course, venom diversity is great, and it relates in part to the kinds of prey that are being harvested by the snake. And there's some evidence that some elements of venom are inducible depending upon the diet. And then there's also lots of evidence that there's genetic variation across the range of various species in venom composition. But I'd have to defer much more detail than that to somebody who's really, really studying it.
Yeah, and so when you say inducible, you mean like people think of, it's different. It's not a venom, but like poison dart frogs, they get that poison from their diet. So when you say inducible, you're saying that some of the variation in the venom has to do with the diet. Was I following?
Yeah, I think that there's good evidence for that different diet types. For instance, if you have a generalist that's feeding on lizards versus mammals, a great example, and I'm not saying that anybody studied this, but my rock rattlesnakes in Big Bend are at least one of my sites were generalists. When you looked at their fecal material, you would find evidence that they've eaten a lot of lizards, but also mammal hair. So they're eating ectotherms and endotherms, and if you find them in a situation where they're eating primarily ectotherms, that may induce the composition of the venom to be different than it is when they're eating mammals. But again, there's some really nice studies out there that show that kind of phenotypic plasticity in venom that sort of comes with respect to the diet. And there's also some neat studies on predator-prey relationships that relate venom compositions to arms races between predators and prey out there.
Yeah, those are fascinating. One interesting thing I've always found with venom is, you talked a lot about, how it's, an adaptation for feeding. It allows the snakes to kill these prey items. It's also, some of this work that's come out that show that a lot of the snakes are actually tracking the chemical signature of their own venoms to find the prey items that they've killed. That always just blew my mind. But it makes a lot of sense, right? If you're a snake out in these landscapes, a timber rattlesnake moves through the forest, there's probably going to be rodent trails, all over the landscape. So it's fascinating. And I guess maybe the last thing with venom, and again, I know you're not the venom expert, but what role in timber rattlesnakes does it play in digestion? And what is the general makeup of their venom? I know we've talked about it varies across the range, but what's the role in digestion?
So that's a great question. There have been very limited studies on the role of venom in digestion, and it's not something that I've reviewed in great detail. But what we do know is that we can't detect. So one of the problems we have is that when you look at digestive responses, like digestive efficiency, which is sort of the difference between the energy that's ingested and what the animal actually retains. We call it apparent digestive efficiency. This is a really variable measurement, and it's subject to a lot of potential experimental errors. And the other kinds of things that we measure with respect to digestive function include things like passage times. How long is the food item resident in the gut? And pit vipers, Harvey Lillywhite's done some really neat work showing that pit vipers have this really annoying tendency to retain fecal material for no apparent reason. They'll hang on to a fully digested bolus for months, and there's been various thoughts about why that might be a thing to do, but none of them seem to make great sense to me. But at any rate, passage times are also extremely difficult to measure with accuracy. And then the other thing we look at is what we call specific dynamic action, which is a fancy way of saying the metabolic cost of digestion and all functions that are associated with digestion, including things like costs of growth that are built in, liver intermediary metabolism and things like that. And that is also a highly variable response. When you feed a snake a large food item, it might take them 15 days to digest it completely at a reasonable temperature. If you drop the temperature, it can take longer. And so these are really coarse measures of digestive function. And I think that has probably interfered with having a really clear picture of the role of venoms in digestion per se. I think the primary role of venom is to dispatch the animal rapidly, and there's tissue damage that certainly potentially aids in the mechanical breakdown of the food items, but to what extent that is true, I couldn't say. But what is fairly clear is that the venoms are designed to rapidly incapacitate the prey item, and either by neurological mechanisms like interfering with synaptic transmission or neuron to neuron transmission, or interfering with the respiratory structure, destruction of the epithelium of the lung and things like that.
Great. So sticking along those same lines of feeding in timber rattlesnakes. So first of all, just so everybody knows, timber rattlesnakes are members of the viper family. You mentioned pit vipers. So timber rattlesnakes, like other rattlesnakes, are members of the subfamily of pit vipers. So
right.
When we say a pit viper, what do we mean? What is the pit, and what is it? Just tell us a little bit about its structure and what it functions for relative to feeding in timber rattlesnakes.
Yeah, this is one of the really fascinating things about rattlesnakes in general. They are a group nested within a larger group called pit vipers, and pit vipers are nested within the larger group of vipers. And the thing that sets pit vipers apart is the fact that they have this dark hole in the front of their faces. And if you look at the face of a timber rattlesnake or any other, you'll see the nostril up on the tip of the nose. You'll see the eye. And then down below and between the nostril and the eye, you'll see this dark pit, this hole. And this is just fascinating stuff. The pit is an infrared sensing organ. And if you think about what is infrared? Well, infrared is heat radiation. And when I talk about this to my students in class, I say, how many of you ever had a pot-bellied stove or a wood heating stove in your house? You put logs in there, you get a fire started, and you can see when you have the door open that there's flames and light, right? you shut the door and you can't tell that those flames and light are there. All that light and radiation is being absorbed by the shell of the stove. The stove temperature is rising, and there's this really interesting property of matter, and that's that as its temperature increases, its long wave radiation emission increases. So there's a law out there called the Stefan-Boltzmann law that describes that relationship. The hotter an object is, the more infrared radiation it emits. And the pit is a very, in one sense, a very simple detector. In another sense, hugely complex. But the pit is a hole, a blind-ended hole. In other words, it's just a pit. It doesn't lead anywhere other than a hole. The back of the pit has a single celled membrane, and then there's a space behind that. And neither one of them are fluid filled. There's a reason they're not fluid filled. It's because anytime you add mass to something, it desensitizes it a little bit to heat flux or to detecting of heat. And so this membrane sits at the back of the pit, and it's covered in neurons that respond to changes in temperature, thermoreceptors. And as radiation comes in from the environment and strikes that membrane, it causes neurons to fire in much the way that light striking the retina of your eye causes neurons to fire in your retina. And the really cool thing is that all of that information that's coming in about the infrared radiation that's hitting that membrane is coalesced into a nerve that's essentially eventually analyzed by the same part of the brain that analyzes the visual image. And so this pit organ is just an amazing adaptation for seeing changes in temperature in the landscape. Now, you can imagine this would be valuable in a lot of ways because many pit vipers, not all, but many pit vipers are night hunters. They get out there in the middle of the night and they're hunting. And often they are hunting warm, small mammals. Small mammals are endotherms, and they're generating their own heat to stay warm, just like we do through metabolism. And that heat comes off in the form of radiation, long wave radiation that can be picked up by this sensitive membrane of the pit. So even in the dark, these animals can see in the infrared. And it's been shown, Ken Cardong in his lab's done some really elegant work showing that heat sensing pit is an important navigational tool for directing a predatory strike. So pretty neat.
Yeah, it is just such a great adaptation, and something not unique, but it's just a fascinating feeding adaptation. So
yeah.
I think I'd like to transition a little bit. I want to talk about reproduction, but I'm assuming we can cover some of that as we get into life histories and talking about some of your research. So I do want to segue a little bit, and I want to talk about why timber rattlesnakes is the focus of your research. I mean, I know, as you mentioned, you got a job in Arkansas, and their timber rattlesnakes were there, but I also picked up on you saying that they were of the right size and some other things. So I'm curious to hear about that. But before we do that, I think it would be great to just have you kind of speak, to a minute or two to a few terms, just to kind of orient some of our audience that may not be as familiar with some of your research and some of the terms in your research. So let's start with, you just mentioned endotherm. Why don't you describe what an ectotherm is? And timber rattlesnakes are ectotherms, as so ectotherms as compared to endotherms. And you might touch on, the terms cold-blooded and warm-blooded, because a lot of people have probably heard those, and you can even tell them why we scientists don't like those terms so much.
Right. Yeah. So if you think about the word endotherm or ectotherm, the second half of the word is therm, which refers to heat, right? And so ecto and endo are modifying the term heat. And in this case, it's about the source of the heat that allows the animal to maintain its physiological function. In some sense, everybody realizes that if you cool an animal down too cold, it dies. And if you heat it up too much, it dies. And so, the temperature of the animal needs to stay within some kind of limits, and the limits vary among different organisms. Ectotherm and endotherm refers to two different strategies that are designed to allow organisms to regulate their temperatures. Endotherms are animals like mammals, people, that produce their heat internally. We have internal furnaces that produce heat. They're metabolic processes. Now, every time you burn a molecule of glucose or a molecule of fat, that's a controlled process that releases heat in steps. And at just about every step, some kind of usable heat or some kind of usable molecule is produced. In the case of ectotherms, ecto meaning outside, ectotherms obtain their heat from their environments. And that really is a critical difference between how mammals function and how reptiles function. So, for example, as long as there's enough food, many mammals can stay active through winter months. Well, that's generally not possible for reptiles because they have to obtain their heat from the external environment. So, and you've heard people use the terms cold-blooded and warm-blooded. I avoid those words like the plague, as Chris just pointed out. They're old terms that were shorthands essentially, but the really cool thing is that we might think of fish as being cold-blooded, but there are some really amazing examples of fish that actually have highly developed adaptations to produce heat to warm either their whole body or parts of the body. So it's not a strict dichotomy, warm-blooded, cold-blooded. And I also have to point out that just because an animal is an ectotherm does not mean that it isn't an effective regulator and runs its body temperature at fairly high levels. If you look at desert iguanas, or you look at collared lizards, they tend to have high thermal preferences, and they regulate their temperatures fairly precisely, almost as well as some mammals. And on the other hand, some of the work I did out in Wyoming on small mammals back in the 80s, you could take a deer mouse, and what you'll notice is that on clear, cold nights, they'll pop up out of the snowpack and run, hundreds of meters across the snowpack. We actually put a tiny transmitter that was temperature dependent in one of these deer mice. It was a Minimeter from the Minimeter company back in the day. And we could track the temperature of this mouse as it runs across the snowpack in the middle of the night, and it just plummets. This poor little guy is trying to make tracks across the snow for whatever reason, and his temperature just plummets. So just because the animal is an endotherm, doesn't mean that their temperatures are not variable. So that the idea of cold-blooded versus warm-blooded just breaks down. It's just not very useful as a concept. I don't know if I got all that.
That's good. No, I think that's great. And, one thing I would say, I did my bachelor's and my master's in kind of classic wildlife ecology and conservation programs, which were really great, especially on the applied side. But, when I went to do my PhD, I did it, purposefully in a biology program, working with Dr. Peterson, who we talked about earlier. And for those of you who don't know me that well, I am not a very good detailed person. I'm very good. My strengths lie in big picture, and that's probably why it's good that I'm doing things like hosting podcasts and running nonprofits and designing conservation programs, as opposed to we're working on something very detailed. So But my point is that when I took my PhD, I took, and I believe it was called environmental physiology or I guess I think it was called physiological ecology. It had different
Right.
names over the
Yeah.
years, but basically I took a course that was partially taught by Dr. Peterson and some other people that talked about these types of things, talked about temperature and physiology, and I will say that it's important for understanding any animal, but if any of you out there are interested in especially terrestrial ectotherms, to me it just jumps out at me. But if you're interested in what a snake is doing and why it's doing it when, I mean, you really need to have at least some level of understanding about the types of things that we're about to talk about, and also things we've been talking about like temperature, because it's it just is so important in how these animals function. And again, the point of me saying I was a big picture person is that typically a class like physiological ecology would not be my favorite class, but I will say that it is certainly one of my top three, if not the top class that I ever took. As I was going to college, it was just incredible, and in that one semester, I feel like it taught me so much about reptiles. It not even just taught me something; it gave me this framework. It changed how I think about how they function. And so anybody, the message there is anybody who's really interested in what snakes do and why they do it, I think it's really important to at least get some level of understanding of these types of things. So
I totally agree. I teach physiological ecology. It's a graduate level course, and it's just amazing the depth of that field, and you get right down into the weeds about how the measurements need to be made and what they mean when you make them. It's a very complex field, and a lot of times the detail nature of it goes unappreciated. And I think it's important that people get a good grounding in the theory behind what they're doing, so that they can interpret results that they get in a meaningful way. And so for me, that was my experiences at the University of Wisconsin with Warren Porter, taught me about heat transfer physics and mass conversion and things like that. And then my experiences at the University of Pennsylvania with Art Dunham taught me about the ecological and evolutionary context of those things in a much more detailed way. And I think, that's a really, really powerful. I think, if you look at conservation problems or trying to understand any pattern about animals, the fundamental mass and energy fluxes that happen on the order of that whole organism are critical to understanding whether populations are going to persist or extirpate. And it's all about growth and reproduction. There's so narrow a set of conditions that allows animals to grow on a good schedule, to mature, and to reproduce on a good schedule. Then there's a whole lot of space out there in terms of environmental influence where that's just not possible. So that's key.
Yeah. That's great. So let's shift from that and really get into some of your research over the years. And I want to start by having you give the real brief 30,000 foot level definition of life history, just in a couple sentences, and then have you tell us first of all how you describe the life history of a timber rattlesnake.
Yeah. Okay. One thing I realized, you asked me a question a few minutes ago, and I did not. I addressed the second part of the question, but not the first part. And I think it's important for us to backtrack just a second. You'd asked me about why timber rattlesnakes are such good models for the kind of work that I do, and they are really good models for certain classes of questions. And for one thing, they can be locally abundant and seasonally predictable. We know where they're going to be in the spring when they emerge, and in the fall when they go back to their hibernacula. They have a large body size, and because of their feeding biology, they have a very flexible body cavity. And what that means is we can put a radio transmitter that'll last 2 or 3 years in the body cavity, and it doesn't seem to bother the snake. And that's partly because they eat these giant meals and expand their guts as a matter of course, and they can easily accommodate a small transmitter in the gut without any negative impact. In the body cavity, not in the gut. I need to be careful about that. They're great models for the application of temperature telemetry. And I would say that a lot of what we know about the effect of temperature in the environment, of the thermal environment in ectotherms, comes from work on snakes because of this advance of telemetry. For years, lizards were considered a model organism for physiological ecology, and they still are. But one of the interesting things, if you look at lizard research data on body temperature, is most lizards are small-bodied, and you can't put a transmitter in them. And so the only time you can measure their temperature is when you can get them in your hands, when you catch them. And usually that means we know a lot about their diurnal or daily temperature, during the daylight, but we don't know anything about their nighttime temperatures or temperatures when they're in crevices or something like that, where all we can do is estimate what's going on there. In snakes, we've kind of solved that problem, and it's been a big plus. The other thing is that's cool about them is rattlesnakes are fairly simple behaviorally. And I know there's going to be some behavioral ecologists that cringe when I say that. But the fact is that, you can walk up on a snake in the environment and you can look at what it's doing and what the context is. And you can usually infer what it's doing. Okay. Is it looking for a mate? Is it in the middle of courtship? Is it copulating? Is it laying in ambush, waiting for a squirrel to come across a log top? Is it basking, right? We can categorize the behaviors, and a lot of times they're what my colleague Dave Duvall used to call functionally dedicated episodic activities, which is a mouthful to say that when they do something, they tend to do it for a while for one reason, and then they change and do something else. So they're perfect. They're like dogs in that way. If you tell them to change, they do something different, But anyway, the other thing that's cool about them is for guys like you and me, Chris, they're easy to track on foot. We don't need helicopters or big trucks or ATVs to get after them. We can put a radio track tag in them and we can follow them for extended periods and do just fine with that. There's some downsides to working with snakes. One of them is that they're secretive and there's lots of capture biases. They generally have low detectability because they're excellent at camouflage. And that means that it's hard to know what the population level consequences of thermal biology are. You usually need really large sample sizes and high capturability to estimate population level processes. So we're real good at sort of estimating what's going on with individual animals, but maybe not so much with whole populations. That's a little bit tougher. And of course, the other thing about timber rattlesnakes is that they're species of special concern, or they're protected in most states. The one thing that drew me to Arkansas was not only are they abundant here, fairly easy to find, but they are statewide in distribution, and they are not protected here because they're doing very, very well. So, okay, question of life history. Life history is an attempt to describe the total lifetime reproductive success of an animal. And the reason we want to do that is because that's an important variable that relates to two things. It relates to population dynamics, how populations might be doing on a landscape, and it also relates to the evolution of populations and fitness of individuals with specific traits. So it has evolutionary significance and it has ecological significance. So that's why we are interested.
When you say fitness, maybe just what would be the more layman's description of that?
Yeah, fitness really boils down to the lifetime reproductive output of an animal.
Great.
And it has different components. There are different things in the life of an animal that affect that. And that's really what life history is kind of about, is quantifying those different factors that affect lifetime reproductive output.
So maybe, I was gonna bring you to the timber rattlesnake here and say, how would you describe, the components of a timber rattlesnake's life history? Kind of describe that for us, if you would, and how
Okay,
you
well,
think about their life history.
yeah, the life history thing is a can of worms, and I'll try to start at a high elevation, and I'll get to timber rattlesnakes more specifically. But in a nutshell, when you read life history literature, you find that people are generally speaking about a collection of traits that describe reproductive output. They might be age or size at birth, growth rate, size at maturity, clutch size, clutch frequency or litter frequency, survivorship is a big one. All of those things sort of describe the persistence of the animal and the total amount of reproduction that comes out of it in the course of its lifetime. I've always found the collection of traits definition to be frustrating because people use traits, different traits in different publications. And sometimes those traits are expressed as individual values versus population means. And sometimes and the other big thing is that there's no real unifying theory that pulls it all together and tells you what is a life history trait and what is not. Art Dunham, my PhD advisor, and Bruce Grant and Karen Overall published a really cool paper in 1989, and it appeared in Physiological Zoology, volume 63, number 3. And it's basically about the interfaces between biophysical ecology and population ecology. And it offers a definition of life history that's what I call an allocation-based life history definition. And the idea is that a life history is a genetically determined or heritable set of rules that determines 3 fundamental allocations that animals make. The first allocation is the allocation of time to the different behaviors that they have to do. The second allocation is how they parse out available mass and energy to the different functions they have, like maintenance, reproduction, growth, storage for winter or whatever. And the third allocation is if for a given amount of energy devoted to reproduction, how do you break that up into individual offspring? So, do you make one big baby like an elephant or a person, or do you make 1 million small babies like a sea urchin or something like that? And there are all kinds of interesting things that interact to affect how those rules are designed and how those rules are expressed. One of the things that's important is the environment that the animal lives in. So the environment is going to set some constraints on what's possible. The another thing that's important is how the organism is put together, what their metabolic demands look like, what their growth potential looks like, how much food they need, what kind of efficiencies of conversion they have. All of those aspects produce constraints on the possibility of what those rules can say. And the final thing that's really cool, remember, this all has to relate back to the fitness of the animal, to lifetime reproduction at some point. The decisions, the allocation decisions that animals make about those 3 allocations are subject to survival and fitness in nature. In other words, there's a risk associated with every decision that they make. So as an example, if an animal decides to make 2 or 3 more babies in one given year, and as a result of that doesn't have enough energy to survive the winter, that's a fitness consequence of a decision, of an allocation decision. The opposite of that might be make a few less babies, survive another year, and then make another clutch the year after. All of those decisions affect the total reproductive output of the organism over time. And so I really prefer that sort of unifying allocation-based theory for the definition of life history. And it turns out, if you think about it in great detail, it's absolutely amazing how much depth and how much mechanistic understanding comes out of that. So relative to the timber rattlesnake, we do things like trying to understand the allocation of time and how factors like seasons and weather patterns affect how the snakes use their time. We also spend a lot of time making bioenergetic measurements of growth, measurements of reproductive output, measurements of the metabolic cost, both the resting metabolism or lowest rates of metabolism they have, and also the field metabolic rates where their whole energy expenditure is considered, and storage dynamics as well, to try to understand how the environment is influencing the allocation of those mass and energy packets into different functions that are required by the animal to survive and reproduce. And then we also look at reproductive output, but usually within a species, there's not a lot of variation. Our timber rattlesnakes will have offspring anywhere from 3 offspring to up to 9 to 11. About the biggest litter I've ever seen out here is 11. They get much larger litter sizes in other parts of the range. But we look at that as well and try to understand the factors that influence litter size. So the whole idea here is to sort of remove the abstract sense of life history as a collection of traits that describe the animal to actually a functional model that allows us to understand, the causes and consequences of different functions that happen within the animal. Does that make sense?
Yeah, I'm following it. Maybe you could get into a little more detail, maybe on some particular trade-offs or some particular examples of, say, how timber rattlesnakes deal with some of these allocation issues that you discuss.
Yeah, that's a great idea. So a good example, timber rattlesnakes are what we call low energy systems. They're remarkable at fasting for long periods, and they slowly use up mass and energy. There is a point where they starve to death, but it takes a long time. For example, we have some data on some snakes that didn't feed in the field for upwards of 2 years and still managed to feed and survive that period. So they may be one of the best examples of animals that are able to tolerate food deprivation for extreme periods. On the other hand, when food becomes available, they are hugely flexible and they can ramp up their production in terms of growth and reproduction in very short order. And we've actually measured that by looking at reproductive rates, growth rates among years that differ in food availability. In the Ozarks, where I work, food availability is ultimately limited by acorn mast. So if you have a series of bad years with acorn crops, the small mammal populations starve and die off. The squirrels disappear, the chipmunks disappear, and you can't hardly catch a small mammal to save your life. These snakes minimize their energy output. Instead of making the decision to allocate all their energy into making a few babies and then dying afterward, they go into low energy mode. They allow some of their internal organs to atrophy if they're not using them, especially the organs of the digestive tract, and they minimize their activity and they wait. And when the mass crop changes and small mammal populations bounce back, which they apparently can do very quickly, on the order of a few months, they start feeding again. And it's so cool because at times when that happens, when we have a really low food period, and then suddenly there's a lot of food on the landscape, the female rattlesnakes will actually synchronize their reproduction. Most of them will reproduce in the next year. So it's all driven by that food availability. And the life history part of it is the fact that the animals have this capacity to dump their metabolic costs down to very low levels and wait out these long periods of food deprivation, and then over the very short term, take advantage of a replenishment of the food supply and reproduce and grow and just resume their life histories again.
Yeah, that's a great example, using the, starting with kind of these, the cyclic nature, say, of the acorn crop, and then going all the way to the snake. I often, when I give my timber rattlesnake presentations, I like to go back in time and imagine for the audiences what these landscapes might have been like with the American chestnut. Not completely, but there was there's a pretty strong overlap between timber rattlesnakes and the historic, well, I guess still the current distribution of the American
Yeah.
chestnut, but the chestnut does not produce the nuts, but
Uh-huh.
anymore. But historically, like in the southern Appalachians here where I live, 1 out of every 4 trees would have been a chestnut. So it was the dominant mass
Yeah.
producing tree.
Right.
And on average, they would produce a higher amount of nuts or mass than the acorns, and they would do it in a more consistent rate. So I have no, we can't go back in time, but I just like to imagine what some of these landscapes might have been like with that food source and how it would
Yeah.
have impacted the rattlesnake's life history.
Yeah, that's a great point. And, in Arkansas, in addition to losing all of our chestnuts, I mean, yeah, we've got these chinkapins out there that are these winky little attempts at chestnuts, but they reach a certain size and the blight knocks them back again. They never get any size to them. Anyway, those chestnut trees provided an incredible food source. And the other thing is that there was a lot of oaks here, and they were much larger and had much more predictable masting crops back then. And the interesting thing is that about 100 years ago, pretty much the whole state of Arkansas was clear cut for wood and allowed to grow back without any real management except for fire suppression for the most part. And what the result is that we get this very closed canopy, even age stand of trees that never get any size to them and are competing for light. So if you look at sort of the forest where I work, it's the density of oak trees is unbelievable, and none of them really get any size. So when there's a good mast year, they do fine, and there's a lot of acorns produced. But when there's poor mast years for other reasons, like climatological reasons or fire or some kind of a weather event, there's the leaves on those trees and that closed canopy shut off all the light to the floor of the forest, and there's no food growing for small mammals on the floor of the forest. So you get this problem where the whole system becomes tied to these wild swings in acorn masting crops, which is completely abnormal, right? And what we've done in working with the Arkansas Game and Fish Commission is done some cool experiments where they've gone in and clear cut some areas and burned some areas, and we've kept track of what happens to the small mammal populations and what happens to the snakes. And it's pretty cool when you open up the forest here and burn it. The first thing that happens is all of these prairie plants that are in the seed bank wake up and they grow, and it's like you get prairie plants everywhere, and they're of course seed producing. They produce all kinds of seeds. And the small mammal populations rebound pretty quickly as well. And then we can even detect that snakes that use these areas that have higher ground cover and higher seed production on the ground are improving in body condition over snakes that are in areas that were not manipulated, that are in close canopy. So there's a really interesting connection between sort of the whole food sources in the Ozarks and how this whole thing rolls out with respect to acorn mast. And we continue to work with the Arkansas Game and Fish Commission, because my belief is that what is good for deer and turkey and quail is great for small mammals and is great for snakes. And sometimes people think, all the hunters want is a place to shoot animals, and I don't like people that shoot animals. Well, fact is that if you partner with them, and sure, yeah, some deer are going to get shot. That's actually a good thing. And, you have this opportunity then to partner with them for land, use conversion that benefits all wildlife on the landscape. And, if we did that all over the place in the right way, in the area, with the right information, the right kind of an approach, because it's not one size fits all. But if we did that across the range, we wouldn't be having these discussions about our rattlesnake populations going extinct in specific states.
Well, that's great. That got to exactly where I wanted to go, getting into kind of the applied side of things
Right.
such as fire. And so one last thing I'll ask you, and then we'll transition to wrapping up here. But the last thing I want to ask you relative to your research. So you mentioned I think you called timber rattlesnakes kind of these low energy systems, and you've talked about their life history and the types of trade offs that, allocation decisions that they have to make, and
Right.
how things on the landscape, whether they're natural or human induced, how those can affect and ultimately affect the snake. So, just curious, do you see the rattlesnake's life history or how they function as making them more susceptible, say, to what humans do to landscape, or do you actually see it as an adaptation that allows them to weather or to deal with human changes to the landscape in say a better way than some other species? I don't know if I articulated that well, but it's kind of two sides of a coin, thinking about the human impacts on the landscape, and this goes back to something you actually told me at a conference one. So I don't know if you remember.
So, yeah, you might have to remind me because there's been a lot of beer since then. But
Yeah.
So yeah, that's an interesting question, and my first response or thought to it is that I would not make the argument that the current capabilities of these animals to live in a boom and bust environment has been significantly affected by human activities. What I would say instead is that ability to take advantage of boom and bust sort of dynamics is ingrained in the biology of being a rattlesnake that has evolved in the case of timber rattlesnakes over the last, somewhere like 10 million years on this continent. But on the other hand, what we notice is that, as humans use and abuse resources, in some places, these animals are resilient and they manage to take advantage of, new conditions. So they are still persisting in the Ozarks in good numbers, even though the forest quality has degraded significantly. Well, that's true in Arkansas, and I think it's primarily true because even with all of the problems with mass cropping, we still have enough food out there to support these populations. I will say that we've gone through population bottlenecks where animals have been lost to the study and lost, and the densities have dropped qualitatively. I can't demonstrate that in real numbers because it's very tough because of the sampling bias issues I talked about earlier. But in other parts of the range, these kinds of interferences can be fatal for the population. And it involves a couple things. One is, land use changes that affect food availability on the landscape. And it might be coupled with the advent of high traffic highways through prime habitat that increase snake mortality, road mortality to the point where the population is no longer viable. And so, those factors interact differently on the landscape. The relative importance of different factors that affect all the little aspects of mass energy allocation and risk associated with those decisions, the relatively more or less important factors vary all over the map. Now, one thing I'll say, and maybe this is what you were getting at, is that if you want to have an effective management strategy for, I would go so far as to say pit vipers in general, but certainly for timber rattlesnakes, probably the most effective thing you can do is figure out a way to feed the food items, to feed small mammals and squirrels and chipmunks. If you can modify the habitat to increase food availability for small mammals and they have a positive population response, that's probably the most effective thing you can do to rescue a timber rattlesnake population.
Yeah, great. Well, the thing I was referencing was related to a lot of what you were just saying. But, as I started my doctoral work on the Great Basin rattlesnakes, and we were looking at how this invasive plant was changing the prey. But anyways, I was kind of always using their life history, their reproductive biology, and saying that it was making them susceptible to. And you just kind of opened my eyes to look at the other side of that coin, and just say, hey, we'll keep an open mind here. Think about that life history stretch. Actually, a strategy that evolved for them to weather and to deal with boom and bust type cycles. So, there's not a right or wrong answer there. I just was curious to hear your
there's
thoughts.
Two sides to it for sure, and you're right, because on the other hand, and this is I think a very traditional way of looking at rattlesnake life histories, that they tend to be, not all of them, but they tend to be long-lived, infrequently reproducing organisms. And they generally have high adult survivorship. In good years, our snakes survive at greater than 90% out here. In bad years, bad food years, it can be 50%. And so what I'm hearing you say is what I hear a lot of timber rattlesnake biologists from the Northeast say, which is that, these increases in mortality that are due to road traffic and to human influence of various types have essentially imbalanced the life table, have imbalanced the life history, and made it almost impossible for the populations to grow. And I think that's a very real effect. I mean, when you think about the history of the animals on the landscape, you take a place like Wisconsin, there was a bounty on rattlesnakes up until 1976. And it was I think it was $1 a head, or 50 cents a head, or a tail, or something. And people, through my years of learning about timber rattlesnakes in Wisconsin, I would constantly run into people who would say, I used to bounty snakes off this hill, for years, and now I can't find any up there anymore. I don't know if there's any left. And sure enough, you'd go look and you wouldn't find a single one. And so I think that those human activities can't be, that increase in mortality due to those kinds of activities, and road mortality can't be, that's a vulnerability in the life history of the rattlesnake that I think often drives them into population decline.
Yeah. Great. Well, last thing I want to ask you about the research, and then we'll wrap it up here. And it is a little bit of a shift of gears, but I know you've been involved with this at your university. So, how would you describe the importance of animal welfare when you're going to do the type of research projects that you do? And then just briefly, how do you deal with that in a university setting? What's the structures in place for that?
So, I think every one of us who engages in research has an obligation to carefully consider the relationship between whatever pain and suffering you might cause to an animal relative to the scientific value of the information you're going to gain. And, I struggle with this. There are some things that I feel comfortable in terms of minimizing pain and discomfort. I feel like we can put a small radio tag in a rattlesnake without dire effects. And we've had good success doing that over the years. But there are other kinds of things that maybe I wouldn't do as a biologist, because I don't feel that the information is justified for putting the animal through that kind of an issue. And I think everybody maybe has a different place where they are with that, but there's a whole other issue. And that's that when you're dealing with rattlesnakes, and you have a physiological question you want to answer that's going to cost you, some animals, you're going to have to find some animals. Question is, where should those animals come from? Should they come from field collections? And if so, how should they be field collected? What's the status of the animal in nature? Should we be interfering with them in that respect? Especially if this is going to end up in the death, ecological or otherwise, of the animal. And ecological death is you can't return them to nature anymore, or actual death. And, to me, as I've gotten older, to watch sort of snake populations declining nationwide, I am much more cognizant of asking the questions about the value of the research relative to the use of the animals in research. The other thing I think is really important to note is that the government's policies on animal care and use are based largely on laboratory animal care and use in a biomedical setting. And they don't really cover or cope with the variety of collecting and laboratory and field procedures that most wildlife or animal biologists go through in the field. And so we made some attempts. I mean, I was involved with a redrafting of the animal care and use guidelines for the ASIH back in the early 2000s. And we came up with a decent document, and it's being revised again. We've had a lot of trouble getting in specific IACUCs across the country, that meaning the Institutional Animal Care and Use Committee, getting them to adopt those guidelines as something that they need to use in the cases of field research on reptiles, for example, or amphibians. A few years ago, Robert Sykes at University of Arkansas, Little Rock, and I and Ellen Paul, Bob is a mammologist, Ellen Paul's an ornithologist. We got together and wrote a paper about the application of taxon-specific guidelines in animal care and use. And I stand by that 100%. IACUCs need to take the initiative to learn and recognize that that not all animals can be treated like laboratory rats, and that field research is a different animal, a different kind of procedure. And I think we have a long way to go on that. One of the other things that has consistently concerned me about how we handle animal care and use in this country is the appointment of members to the Institutional Animal Care and Use Committee has to be done very carefully, because we want objective people that are scientifically minded, that understand the pluses and minuses. What we don't want are people that have opinions or attitudes that are from the outset anti-research. And there are a lot of people that are like that. So I think it's I'm probably going to get, crucified for saying that, but the fact is that, we need to always be, and I think everyone could agree that this is a question of balancing the value of the endpoint of the research with the impact on individuals and potentially populations of engaging in it. And I think everybody needs to have that conversation and decide where they stand on that.
Yeah. Yeah. Well, that's just a great way to describe it. So thank you. So we're going to finish up here. As you may or may not know, I'm an avid outdoorsman, and I hunt and fish and travel all over the country to do those activities, and oftentimes sit around campfires with good friends and tell great stories. And so we're starting a new tradition here, because I get as excited about snake stories as I do hunting stories. And so I would love to hear your best snake story. It can be science related. It can be anything. Just love to hear a good snake story.
Oh man, there are so many, because I've been in the field with snakes, most of my life. And a couple, the first time I ever saw a rock rattlesnake was just unbelievable, because I knew there was something live on that rock, but it was so well camouflaged I couldn't figure out what the shape of it was. That's how good it was. But probably the most, the most exciting, most memorable experience I've ever had. About 10 years ago, I went to New Mexico on a brief collaboration with Charlie Painter, Larry Camise, who is one of my current graduate students now, and Andy Holycross. They had a prairie rattlesnake den in an old mine shaft out on some private property, and that they were working at. And we were going to do some sampling in the dead of winter. And I kid you not, this mine shaft, cuts down into the hill at about a 40-degree angle, and it's narrow. One person at a time gets through there, and I don't know, maybe 40, 50 feet deep, and the den is at the bottom. And they had marked hundreds of snakes in that den, and we decided we were going to go down there and collect some rattlesnakes. And it was the most surreal thing. First of all, it was snowing that day. So we're going on a snake hunt, and it's like almost blizzard like, but not quite. And we're scooting across the prairie on these ATVs to get out to this site, and the snow is whipping in your face, and it's cold and everything. And you're thinking, are we out of our minds? We get to the opening of this thing, and there's just the rocky black hole. It's an old mine shaft in the middle of the prairie. And, we climbed down into this mine shaft with a flashlight, and I think they sent me down first, but I don't remember if Larry went ahead, but there were two of us that went down. And as you're going down into the shaft, I mean, first of all, there's a small prairie rattlesnake under a crevice right at the opening, of this thing. And whereas we're climbing down, looking into crevices on the side of this thing, you can see rattlesnakes coiled up in there, We get to the bottom, and there's kind of a little small opening there. It's a backfill where they closed off the deeper parts of the mine. And I'm like, where's the snakes? And Larry points up, and I look straight up, and there's this giant crevice arching up over my head. And that thing is packed with live prairie rattlesnakes hanging right over your head. And I was like, oh, this is the coolest place I have ever been in my life.
That sounds great.
This is unbelievable. And so we wanted to get 30, 40 animals for a little winter sampling we wanted to do. And we were trying to look at water dynamics, but it really didn't work out very well. But long story short, we had to catch these snakes. And so we're reaching up with snake tongs, getting a hold of the animals and literally pulling them down onto ourselves into a bucket in this crevice. It was the wildest thing I've ever done. I think it was safe enough. They were really good at hanging onto the ceiling, and we were careful about not to drop anything, on anyone, but it was kind of a rodeo. And the whole, as soon as we started, pulling those snakes off the ceiling out of that crevice, they started receding into it. And so the whole thing was over in 30, 40 seconds. And we probably had captured 25, 30 snakes.
Oh, wow. That's good.
Yeah.
Yeah.
It was just an incredible experience. I to this day, it's still one of the most exciting, and insane snake experiences I've ever had.
That's great. Well, thanks for sharing that. That is a great story. So, well, we've gone well over an hour here. So, I just wanted to thank you, Steve. It's great to reconnect with you, and it's been great to do a small part kind of sharing your story and some of your research with a broad group of people. So if people are interested in learning more about your research and other things that you're working on, what's the best way for them to do that?
Well, I've got a university email, and there is a brief website up there. I used to have a much bigger website, but university conventions have streamlined things. But anybody can send me an email if they have questions. SBEAUPRE@uark.edu. And I'm happy to try to answer them. It's sometimes difficult for me to get back to people fast because I've got a lot of stuff on my plate right now, but I will do my best. And, I think my CV is posted. They can see some of the papers that we've done and some of the different kinds of projects that we've done. And I like to say that it's been just a joy talking to you today, Chris, because, we share this history and I know we're on the same wavelength in a lot of ways with respect to these animals. And I appreciate, so much the work that you do and the work that, the Orient Society does. And, I have always thought what a great career path you've taken. And, lately I've had a lot of conversations with people who are real hardcore active conservation biologists. And, I look back on my career and I think, I've answered a lot of questions. I've generated a lot of questions, and it's been a really interesting romp. The question is, what's the end result? The end result is I become more motivated to become more active as a conservationist with respect to these animals, and thinking of ways to partner with land owners like the Game and Fish Commission in Arkansas, the state Heritage Commission, to try to make it a win-win for everybody. So I think I urge people to think in those terms, and then go where your nose leads you.
Yeah. Well, great. Well, thank you, Steve.